This is TWIS, This Week in Science, episode number 911, recorded on Wednesday, February 1st, 2023. Will the dodo fly? Hey everyone, I'm Dr. Kiki, and tonight on the show we will fill your head with Bigfoot, dodos, and fungi. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. A giant green comet is passing by Earth today. One that has passed by the Earth before, some 50,000 years ago. We only mention it because, like so many things we talk about in the following program, it's just really neat. There are a lot of neat things on planet Earth, not the least of which is you. You in the overall generalized sentient hominin sort of way. Specifically you is no doubt very neat and interesting and multifaceted in your particular way of existing, living on a moist, rocky planet with a molten lava core orbiting a nuclear fireball like you do. But just keeping it generally human for the moment, what a wonderful thing that is to be. Sure, there's the problems usually caused by other humans, but there's also all of the solutions, often created by other humans. Not to go so far as to make a point about anything here, but there is something to be said for having the ability to think and to do. Without it, I can't even imagine what there would be to talk about. Humans have brains capable of understanding just about any concept. And we think that's really neat. Which is why we have brought you a selection of interesting concepts pulled from the scientific research of the day for you to wrap that really neat brain of yours around here on This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back to talk about science. Yes, if you are a fan of the show, you know it's because of the science we're here for. We're here for it every single week. I have stories this week about asteroid asymmetry, some fun for the fungi or fear? I don't know. A big map and a little bit about mitochondria and alcohol, but no, those aren't the same stories. Justin, what did you bring?
I've got Bigfoot meets big data, squid pains, hydrogen upgrades, and Neanderthal skulls.
Lots of Neanderthal skulls everywhere. Blair, what is in the animal corner?
Yes, I have dodos. I have jet-lagged fruit flies. And I also have Bigfoot.
Well, too bad I got to say what stories I'm doing first on the show, and therefore that's how you.
So
you get
is
there
it
first.
that you have a Bigfoot and you have a Bigfoot, so together you have big
I think we're doing this one together.
Yes.
Because it turns out that something I did not know about Blair is she's a huge Bigfoot aficionado.
Yes, I am.
Learn something.
Well, every once in a while we have to go down that path of
Cryptozoological.
Yes, that's exactly where I was going. We're going to head down to the crypto zoo. And, well, not crypto, but you get it. You get it because you're here for the science. All right. As we jump into the show here tonight, I want to remind everyone that if you have not yet subscribed to TWIS, please do. You can find us as a podcast, places that you find podcasts, and you can find us streaming live weekly on Wednesdays at 8 p.m. Pacific time on Facebook, YouTube, and Twitch. We are TWIS Science on Twitch, Twitter, Instagram, and Mastodon on the Universidon server. And if you head over to TWIS.org, you will be able to find all sorts of things, links, show notes, and ways to subscribe to TWIS. Okay, enough of that. Let's jump into the science. Have you ever wanted to know where all the matter is?
I mean, it's all around us, isn't it?
I mean, you look at the
But it's
astrophysicist,
missing.
right?
It's
Yeah.
so much of it is missing.
So
We don't
much
know
is
where
miss-
we put it.
And so this is the thing. You don't necessarily go, what's the matter, to an astrophysicist. You say, where's the matter? Because they don't know what all the matter is yet. So don't ask that question. But thinking about where all the matter is, researchers have taken data from a couple of very large telescope surveys, the Dark Energy Survey, which was surveying from a mountaintop in Chile over 6 years, and the South Pole Telescope that looks at traces of radiation from right after the Big Bang. And all of this data was combined and put together to make a picture of the universe so that we could get an idea of where both regular matter that emits and reflects light is, and also dark matter, which is matter that's not visible, but we know it's there because it has mass. It has a gravitational impact. And that's exactly what these researchers have done with the data that they've collected, is they've been able to take all of the gravitational effects, take all of these surveys together, stick them together, and give us a picture of where in our view of the night sky there is matter.
Oh,
And this
gosh.
is-
And according to that picture, the entire universe is a Dalek. Doctor Who.
The entire universe, yes. So we get to understand how matter is distributed, where it's distributed, and then we can start to associate that with more visible data from these telescopes that we have now looking at the skies to be able to give us an idea of what is causing the various amassings of data in different places. So they're using things like gravitational lensing to be able to determine when things are layered in space, really, because they're behind each other and not necessarily all lumped in one spot in space. But the researchers say that all of the data, it all seems to fit so far. The amount of data of matter they expected, the distribution of matter, how it all is fitting there. They're like, oh, this is great. It all fits together. This fits the puzzle of how we think the universe should be working and how it should be put together. And then, though, they're like, it's not messy enough. So an astrophysicist from the University of Hawaii, Eric Baxter, has said in a press release that it seems like there are slightly less fluctuations in the current universe than we would predict, assuming our standard cosmological model anchored to the early universe. So if we were to take the early universe as the starting conditions, and then based on our understanding of math and physics and everything, and extrapolate outward, then there should be more clumps. And the universe is not messy enough. It's not clumpy enough. It's kind of neatly spread out, according to this new survey. So now, this offers an opportunity for other researchers to be able to do different analyses of the data, to use different telescopes to be able to take a look at these views, and to be able to figure out, are we looking at it from the right angle? Is there something else that we should be using to be determining whether or not we have a good perspective on how the universe looks and should look?
Yeah, it's really tricky because all of the knowledge we have of the universe is based on our observations from this one perspective.
Yeah.
And then dialing it back, sort of back engineering it to how it could have started. So yeah, our perspectives, of course, are very limited from this little moist, rocky planet, trying to figure out how the entire universe formed.
I know.
And the fact that
We have one view. Like, we're not even like, oh, let's triangulate from multiple perspectives in the universe to be able to get idea. Nope, we're just looking from one space and just jumping to conclusions. So, yeah.
As we do, and as we must, because we have no other choice.
We have no other choice. Send
I
the
do like that.
robots.
There's something fun about the universe being less messy than we expected. You know, if it was more chaotic than we could have imagined, then well, you know, you're missing a whole bunch of chaos in this. No, actually, it's a lot more boring than we thought it might have been.
Yeah, our universe just needs to be more clumpy. Everybody, get to work. Let those universal dust bunnies build up a little bit more.
It's like when you're a kid and you find out that after you go to bed, your parents just go to bed. They don't stay up eating banana splits all night like you imagined.
No, they're tired too.
Yeah.
In fact, they wish they could go to bed before you go to bed.
Very often. But speaking of dust bunnies, sometimes those dust bunnies look like things out there. And sometimes when people are out in nature, they see things and they're like, it's the Pacific Northwest. It's got to be a Bigfoot.
Yeah.
Justin?
So this is part of the infrequently recurring segments. Did we really need a study for that? In which the answer is always yes, even though the results are maybe what we thought they might be what we expected. So this Bigfoot meets big data. American lore has long claimed a large hairy hominid is roaming deep within our forests. The cryptozoological creature called Bigfoot or Sasquatch has been reported all across the United States and Canada, but most often in the most densely forested areas. Thousands and thousands of sightings dating back many decades have been reported. Over the years, there have been genetic microscopic analysis of supposed hairs, feces, other specimens attributed to Sasquatch found out there in the wild, which after analysis, none of which have been attributable to any unknown creature. Despite the advent and expansive use of digital cameras, including personal cameras, phones, wildlife motion detector cameras, no definitive still image or video of a Sasquatch has ever been recorded. And yet, the legend persists. Okay. So there's a study likely to make some news cycles this week that finds Sasquatch sightings correlate significantly with black bear populations. In fact, what is it? Blair, you also like this say there's one Sasquatch sighting approximately for every few hundred bears in the region. And you could actually predict a lot of Sasquatch sightings just by knowing how many bears there are in an area. So based on the statistical considerations, the study asserts that it is likely bears that people are seeing and not Bigfoots.
That seems fair enough. But yes, Blair, what do you think about these correlations?
So, as someone who's worked with black bears and also someone who's visited many Bigfoot museums, some of this makes sense and some of it doesn't. And so, I don't claim to believe that Bigfoot exists because I don't. But if you look at some of the detailed quote unquote accounts, some of this works very well because like black bears like to move around on their back legs sometimes and they look awkward and kind of weird and they kind of swing their arms back and forth. So in that way, it totally makes sense. Some of the favorite Bigfoot stories that I've seen have to do with kind of leaving things for Bigfoot and Bigfoot bringing you rewards and having, these people who live in the woods and cabins having exchanges with them where they might leave some graham crackers out and Bigfoot brings them something shiny. There's lots of animals that do that too. Definitely a black bear could be doing that. That's totally possible. Also crows, right? But some of this didn't seem right to me. So one of the things was that they claimed that this makes sense because black bears can be similar to the size of humans. Similar is a stretch there. Black bears vary in size a lot, but there's not many of them that are big enough to be confused for a human. Maybe from far away. But isn't that Bigfoot's whole thing? Is that he's like 7 feet tall? There's no black bears that are 7 feet tall when they're standing on their rear legs. So this is a pretty big problem for me. Also, all of the fuzzy photos and things like that do not look like bears. They look like humans, which obviously the likelihood is that they are fake and created by humans, right? But it's.
When you, I gotta pause.
Many of them have been shown to be actual fakes and people.
I have to pause for a moment.
Yes.
When you say all of the, now granted, I'm not an aficionado, but I did a little Google searching for best images of Bigfoot.
Yes.
And I didn't find anything that I would consider even a fuzzy image that I would interpret as a 7-foot hominin. That's nothing.
Yeah.
And the eyewitness accounts of a 7-foot anything is, I'm sure, from a great distance in the deep woods. Mm-hmm. And like, you're not, nobody's taking out a measuring stick.
The foot indentations could very easily be the rear feet of a bear.
Yeah, and a lot of them also have been found to, like, some of the best ones have also been found to have pores on the bottoms of their feet, which also, these big pores on the bottom of the feet would happen if you had latex in a mold that had air bubbles.
Yeah. Also,
so,
the calloused foot pads of things like bears also kind of have cracks and dimples in them. So that would be more likely a result of that as well. So basically what I'm saying is, I believe it's possible a lot of them are black bears. But not all of them. I think you have to remember, you have to remind yourself that some of the Bigfoot lore is fabricated.
Some?
So that's what I'm saying. Some of it could be black bears. The other stuff is just made up. And you have to kind of include those in the conversation because there's these other pieces of it that I think are that don't fit perfectly in the bear kind of.
And there are nature myths, and there are stories that go back, as people in the chat, various chat rooms have been saying, in different cultures around the world. There's Yeti, the Bigfoot. There's, a number of these large, hairy.
One of my favorites, the
creatures.
Rougarou, which is in Louisiana.
Right. So there are myths that they stem from, and these could be animals. These could be other sources, but then we perpetuate them. And people are also out in the forest, and maybe they're scared and they see something shadowy on 2 legs going after, off in the distance. I would run away. I would just, I saw Bigfoot. No, I would more likely think it was a black bear. But there.
Well, this
the human psyche wants to believe. Yes.
This week also brought us a cartoon bear face on Mars, which, like the past human face on Mars, just illustrates it, that human imagination can often be the first thing that we use to see things with.
Right.
When looking at an otherwise ordinary world. I did notice that there were some areas on that map of the correlations that had no bears, but still had, not Pacific Northwest numbers of bear sightings, but did have of Bigfoot sightings.
Yes,
did have Bigfoot sightings. Florida, for instance, which doesn't have any bears, but does have.
Well, so they might be counting the Rougarou or something like that, like a swamp monster in that. Then.
Well, they're looking for Bigfoot. There's Bigfoot.
Yeah, so they might be counting
big hominins.
Yeah, I don't know.
Florida does not have bears, but it does have an active population of Floridians.
It does have an active population.
It also has alligators, which
human behavior. Because
alligators can act weird and rustle things and climb trees and do weird things. So.
No, I think the big overlap with the the bear populations and the high number of sightings and some of those little clips of videos that I did see that did look like something furry moving around in the far distance looked to me like a bear.
Yes.
And interestingly, they didn't correlate this positively with brown bears. I don't know if that's brown bears don't do as much hind leg
There's
walking.
almost none left in the United States, so that's
there's not that
probably
many
why.
of them. And where they are too, I figure is less populated areas with humans and probably people who are a little bit more attuned to surviving and understanding nature if they're living out where brown bears still exist.
Well,
and are less likely to assume Bigfoot because they're more concerned about a bear.
Well, you have to remember also, though, brown bears are, without a doubt, so bear-shaped. They're rounder. They're less awkward when they're on their hind legs. They have these huge round heads with huge, big, cute, fluffy ears. Don't confuse it.
Like we saw on Mars.
Yes. But so that's the
yes.
other thing, is that if you see a grizzly bear, you know that's a grizzly bear. They also don't walk around on their hind legs doing stuff like this all the time and like batting at stuff. Black bears
behavior.
are goofy.
Yeah.
Black bears are so silly. And I really think that's the piece of this that really made sense to me, is the behavior of black bears fitting into this weird, erratic forest man idea. The reason I love Bigfoot is that the beautiful lush forests of the Pacific Northwest feel like a magical place, and they feel so separate from urban life.
Yeah.
And I just like the kind of mysticism of keeping it its own space, kind of separate from human civilization, because it is magical in its own way, in like a weird, I don't know, like,
if there's anything that could be left over from a time before modern society started to impact everything, like
and it's a place where you can really feel submerged and feel separate, and it's I just love the added lore of it, knowing that it's, you know, it's all made up, but it's so fun to immerse yourself in that and kind of imagine that you're in this magical place, which I love. But all that to say, I totally buy a lot of the sightings are black bears, but I do not believe that the falsified evidence is black bears.
David in the chat room is pointing out that Florida does have black bears. It's very small population compared to elsewhere for the number of Floridians that claim to have seen a Bigfoot. But I think still, you know, maybe the rate of Bigfoot sightings go up also when you have just some black bears, because then you aren't used to seeing one, don't expect them.
Sure.
Yeah. Well,
I like the idea that the magical temperate forest being, but we should have a Bigfoot preserve.
let's preserve nature. Yes.
We should have Colossal bring them back from extinction.
Oh, we'll get to your story in a bit.
We just need to get some DNA.
We'll get there. We will get there. Let's move out into the solar system for just a minute. Again, looking at space, but looking at our space, close to home. One of those big, lovely planets with rings out there. Jupiter. We love Jupiter. And Jupiter has a bunch of asteroids that are in its orbit. And people have thought historically, oh, look at all those nice asteroids. They're in the Lagrange points. Oh, look at, why are they lopsided? Why are there more asteroids in this one Lagrange than the other Lagrange? And so a team of researchers has been taking a look at these Lagrange points in Jupiter's orbit, and they're proposing, based on solar system dynamics, that once upon a time Jupiter didn't live where it lives now, and that our solar system, we talked about this on the show before, but that at one point in time, the gas giants were closer in, and everything maybe has moved around, and because of that, we've ended up with objects in places where we wouldn't necessarily expect them based on just the location of everything right now as it is. So this is kind of similar to the first story that I brought, which is we're looking at everything right now and trying to keep an idea of, okay, how did it get there? What's our hypothesis about our solar system? How did it form? So the researchers have determined that there were different evolution paths for these asteroid groups that exist in the L4 and L5 swarm Lagrange points in Jupiter's orbit. And this asymmetry has come about specifically because Jupiter used to be closer in, and at one point it moved. And so the gravitational pull was disrupted. It got booted out into the outer solar system at some point. And what they're calling this is the grand tack hypothesis. This isn't the first time that anyone has come up with this, but it might solve a bunch of problems about how we look at the distribution of matter in our solar system. And it also solves this asymmetry with this particular for this particular idea. So the asymmetry happened when there was an outward migration of Jupiter as well. So Jupiter moved out and then it moved back in. And so you end up getting this, excuse me, higher population of a certain kind of asteroid called the Greek asteroids within one of those Lagrange points. So it has Greek L4s, Trojans in their L5, and there's more Greeks than Trojans because Jupiter moved. It's just another story of how things aren't static. We're just a moment in time, and we think things are the way they are, but they're not.
Still shot.
They
picture
feel like
frame.
they're static because we are a blip.
A blip. Yes.
And if we start
It doesn't make me feel insignificant, I'll tell you what.
I love that, but I love that kind of thing. It's like, oh, insignificant, life is meaningless. No, oh my gosh, there's so much more out there. So much significance to just the fact that we exist as this blip. But anyway, let's move on from blips and asteroids to, you want to talk about squids? Is that what we want to talk about right now?
Well, this is what I would call the best solution so far. When looking for ways to reduce the carbon footprint of human activity, one of the biggest targets out there is buildings. Taken together, not just one building, but all of them together. Buildings consume about 32% of the global energy supply, 50% of the electricity consumed globally, around 25% of the greenhouse gas emissions are used in buildings, in the heating, cooling, lighting of our modern cave dweller lifestyle. That is, by the way, more than manufacturing or transportation. It's just keeping the buildings warm or cool or well lit. And it's going to get worse as the future brings more humans. There will be more buildings required, and global warming increases the fluctuating temperatures, requiring more cooling and heating of the existing buildings. University of Toronto researchers have found a novel approach that will reduce the energy consumption of buildings by about 43% over the best existing technologies, moving us much closer to that net zero goal.
I like that.
Yeah, and they did it by replacing traditional windows with squid skin.
Wait, what?
Squid panes.
Tell me more.
Lead author of the study, published there, the Raphael K, says the key is controlling the solar energy that enters buildings, which we talked a long time ago about Earthship biotexture, which is a completely off-grid building system and very much focused on controlling solar energy.
Right.
There are systems already in existence that can block light, very primitive ones. You can have curtains, you can have blinds, you can have automatic blinds that open and close based on the temperature of the interior of the building to try to add a little heat or block a little heat to make the building more efficient. You have electrochromic windows that have tints in them that can automatically darken, kind of like automatic sunglasses. If you go out into the sun, the glasses will darken in reaction to the light. But these systems don't discriminate between different wavelengths of light, nor can they control how that light gets distributed spatially. Excuse me. According to Kay, sunlight contains visible light, which impacts the illumination in the building, but it also contains other invisible wavelengths, such as infrared light, which we can think of essentially as heat. In the middle of the day in winter, you'd probably want to let in both, but in the middle of the day in summer, you'd just want to let in the visible light, not the heat. Current systems typically can't do this. They either block both or neither. They also have no ability to direct or scatter light in beneficial ways. The system developed by Kay and the University of Toronto team leverages the power of microfluidics. The prototype consists of flat sheets of plastic that are permeated with an array of millimeter-thick channels. So they can pump fluids through these little channels with customized pigments, particles, and other molecules that can be mixed into the fluids that control what kind of light gets to pass through them, such as visible light or near-infrared wavelengths. And they can also control to some degree which direction that light is then distributed. So you can get light further into a building or block more light as you may desire. The sheets can be combined in multi-layer stacks, with each layer responsible for a different type of optical function, thereby controlling intensity, wavelength, tuning, and scattering, transmitting light indoors how they like. So tremendous amount of control can be achieved. You can let the heat in, and the light, you can block some of that heat, but still let the visible light go everywhere, direct it further into the building, all this sort of stuff. So this is a great optimization of solar energy coming in.
Yeah, really, really interesting, taking the inspiration from the way the skin of squids works.
Yeah, so in a few species of squid, their active camouflage is a pigmentary layer of chromatophore organs and a more structured layer of protein cells. And where those overlap, they can together mediate color, spectral reflectance, and spatial patterning of the light. So it's really an amazing sort of piece. The same team has previously worked with the camouflage from chameleons and the sort of crystalline structure of their skin, which can selectively reflect light through, again, a multi-layered tier, which they're using for facades. So in combination, you have windows, squid-based, which can control how light is getting into the building, what heat is or isn't getting in, and a skin of a building that can control, if you want to keep the building cool, you make it a very light color. If you want to absorb more heat, you can darken the surface of the building. You can, in an emergency, you can make your building look like an innocent tree. Forest.
We're only a tree here. It's all fine.
You could theme it up for the holidays. You could actually do some really fun stuff with it.
So, Justin, I have a follow-up question, though.
Yeah.
Doesn't it take energy to push fluid through these windows or manipulate the crystals in the facade? And does that play into the 43%, or is that a separate piece of this that makes it maybe less efficient than it could be?
So they say here, if they had just one layer, this is KN, if it was one layer that focuses on modular transmission of near-infrared light, not even getting to the visual, they could save about 25% annually on heating, cooling, and lighting energy over the baseline. If they had two layers, that gets into that infrared and visible, it's more like 50% reduction in that building's energy consumption. Right. And that's over systems that have automatic blinds and chromatic window.
Right.
So,
right, but so my question is, does it take energy to manipulate?
of course it does. Of course it does.
Right.
But that's the point. So they've done the comparison not off of a building that didn't have a system, because the chromatic windows and the automatic blinds take that same realm of energy. So they've compared themselves to systems that are already using energy to manipulate the solar energy. Right. But yeah, this is pumping microfluids through a pane. It's not a heavy energy expenditure that's taking place.
I'd just be interested to see, because is it equal to moving an automatic blind, or is it more? Probably, is it less? Or
well, they did.
So say it's the same. Say it's a little bit more.
Yes.
Say if it was even a little bit more, twice what it takes. That's still nothing compared to a compressor running a building-wide air conditioning unit.
Sure.
That's nothing compared to a furnace heating a building that the solar energy passively could have taken care of. So, but that's, I think, why they used their comparison against existing systems that are already using some
energy. So you have something to compare it against. Yeah. With how this would work, I think the big question is, how much would these cost in the end production? How much manufacturing could actually take place? Would construction actually adopt this? How can we get the government to add regulations that or stipends that help to add these kinds of windows to buildings? There's so many questions related to the affordability of this kind of thing, because it's going to be more expensive than just a regular window.
Well, that was my other thought too, is like, do you have to replenish the fluid? Like, does it biodegrade over time as a squid body might? I got to refill my window fluid.
Yeah, you might, and we are talking also about the research phase of proof of concept.
Yes, of course.
Right?
Yeah.
So is this still something that we want to do in panes of plastic? Could we convert it to panes of glass that might be more stable in high wind situations or something like this? So there's all sorts of, but the
and the cost savings thing is, though, the energy saving, long-term cost savings it
does look to be scalable. The materials all use the microfluidic material. All of this is very cheap. These are all very cheap. It's a lot of good science behind it, but it's cheap materials in the end. It's sustainable. It's a sustainable solution that directly reduces energy consumption at the point of use, which is also all the efforts we're making to create an alternative energy that isn't polluting. Here's one that just says, whatever you're using, we'll leave that aside for the moment until you figure out the 40-year away plan for fusion or whatever you're dreaming on. Let's just figure out a way to one of the biggest sources of energy, cut it in half at the point of use by using a giant nuclear fireball. It's right there, always on, and controlling how that energy is used or avoided.
Yeah, there are always.
I think it's the best idea I've heard in a long time.
I think it's a great idea. I mean, this is the kind of thing, it's going to change the reflectance of windows. So skyscrapers are going to have fewer birds fly into windows. You're going to have,
that's a good one.
Big buildings. This is going to impact the overall energy outlay. It's going to change the amount of, if it's air conditioning or heating, this is carbon dioxide usually consuming processes, or not consuming, but releasing processes. So I don't know. It seems great. There's always trade-offs to things, but this is the kind of solution that it can have an immediate effect.
And it has an economic benefit, which they're not talking about directly in this study, because they're thinking about the energy consumption as a carbon footprint in the telling of the story. But if you were to tell any business that their headquarters, that $30,000 a month electricity bill or whatever it is, could be cut in half. You know what? Let's that might be worth looking into. And then it's every building. So one of the things I like about this too is to some extent there's retrofitability of existing buildings.
Right.
You don't have to
if it's a film,
gut the infrastructure.
Right. If it's a film that you can apply to a window as opposed to install a whole new window, then that is a game changer. You have teams of people go in, add the film, it changes everything. Instead of getting your curtains, you get squid skin.
Squid skin
for businesses.
Squid skin for the window panes, and then chameleon skin for the building facade.
Yeah.
The future's going to be way cooler, I think, looking than anybody.
So colorful. Oh, yeah. Let's talk about the future. The future, it is going to get hot, and climate change is going to change those bugs that are going to impact our health. And while we have been focused on bacteria a lot, we've been focused on viruses as a result of this pandemic we've been living through, and we're like, oh no. The things that we don't talk about very often are the fungi. Those fun guys. Yes. New story out of Duke University School of Medicine has determined that, and we've talked about stuff like this before, Cryptococcus deneoformans. It's a pathogenic fungus. It's going to make it better at being pathogenic. It's going to make it better at living. They tested it at high heats, and they've published their research in the Proceedings of the National Academy of Sciences that it's going to make the fungus have more jumping genes, and they're going to get up and move around, which is going to lead to more mutations, and there's going to be changes in the regulation of all of these genes. And the heat stress of adapting to the higher heats, the researchers say, is going to be the big issue for speeding up these mutations. And the thing is, from a public health standpoint, it's harder to control because it's not like, oh, we got to keep the people away from each other so the people don't give each other the viruses. It's not the thing that you breathe on each other. Fungi are all around us. Their spores are in the air. We inhale them. They're
in our blood.
Yes, the fun guys are always around. Those fun guys. And, a lot of people might be a little bit more aware of fungal infections right now because of the TV show that's very popular at the moment, The Last of Us. Are you watching it, either of you?
No,
I'm aware of it.
I'm aware of it, and I don't know if I can watch it. I mean, it's a zombie show.
Yes.
There is a fungus, which is a fungus that we've talked about also on the show. It's like, fungus that might impact intelligence and other things.
It's like the fungus that turned the bees into zombies, basically. But
bees, ants, crickets.
Everything.
There's a lot of zombies in nature. That's one of those things that you have to pause and just like, oh yeah, and zombies aren't real. Actually, there's many.
You look at arthropods.
Many examples of zombies in nature.
Oh dear. So
not like Bigfoot.
Fungal infections are increasing because we have a lot of people who have weakened immune systems, or very often because of the pollution in our cities and other places. Also because of underlying health conditions or genetic disorders that predispose them to having weakened immune systems. Pathogenic fungi are increasing because of the warmer temperatures, and they're probably infecting more people. There's more mutations taking place. We really need to be keeping an eye on the fungi.
Yeah,
we do.
And that old adage, "The enemy of my enemy is my friend." We have to go find a bacteria that has formed a resistance against the fungi and create an antifungal based off of bacteria. And if the bacteria get out of hand, well, we got to go find a fungus that's got an antibacterial property. It's just endless. It's just endless once we start down this road.
So this endless road, well, we may have a new vaccine, actually. The University of Georgia has developed a possible vaccine for fungal infections. Dun, dun, dun. So our pathogenic fungi are starting to become, they're getting their drug resistance and all that kind of stuff, just like the bacteria. So, that's no good. It's because everybody's got myconazole or something in their medicine cabinets. But this University of Georgia study they've published in Proceedings of the National Academy of Science Nexus, they have determined that it is able to, they have designed it to protect against the 3 most common fungal pathogens that are responsible for more than 80% of fatal infections. And they looked at 4 preclinical animal models, including non-human primates. And they were able to determine that it showed broad cross-protective antifungal immunity in those models. So they're moving forward to future clinical trials. So this vaccine is targeted at Aspergillus, Candida, and Pneumocystis. And so who knows? Perhaps in the future, we will be trying to get people to pick up a little bit of vaccine acceptance for not just the flu or RSV or COVID, but also for
those are viruses,
right?
So,
right. Those
send
are
your
all
immune
viruses.
system to the gym. Get them worked out for all these different things.
Okay, I don't want to get all anti-vaxxer on everybody.
Oh God!
But I might end up being one with this. So here's for instance. We're going to give you a vaccine that protects your body against bacteria of several order. Oh, that sounds nice. But wait a second. My entire gut microbiome is made up of bacteria that I need to function. And
these aren't bacteria. These are funguses that are pathogenic. And this is not targeting the non-pathogenic
it's not
fungi.
Targeting.
Yes.
I'm just saying we also have fungal microbiota.
We do. Absolutely.
And we have fungi that are doing work we don't quite understand yet as they get into our blood system. They may not all be pathogenic. Now, I know this isn't blanket protect your body from all fungi. I know they're trying to be very targeted here, but this is one of those that they better do. Was my waggy finger thing is happening. A lot of research before they try that out in any human model.
So, I mean, there are vaccines for bacteria currently.
Yes,
so
there are.
Tdap. Tdap is all bacterial pathogens, right? So that's part of the process. Is making sure you're not killing the
Make sure
stuff,
it's the targeted is the
part
important
of the process.
thing.
Yes.
Yeah.
Targeted is the
part
whole
of the recipe,
aspect of
always.
Yes. And as we have seen, there are a lot of questions that are not just what are the fungi doing on their own in there, but also how are they interacting with bacteria? Because there have been some studies that have shown that the interactions between different fungal species and bacterial species have metabolic effects or change the way that drugs are metabolized, or there are lots of interactions that still need to be taken into account. But when you're talking about pathogenic fungi that are really causing disease and hardship for a large number of people, you start to get to a point where it would be great to have a vaccine. In the meantime, until we hopefully come up with some other great antifungal strategies, we need to help our immune systems, and we need to be concerned about the fact that the warming world is going to be changing the way that these microbes evolve with us, and it could cause them to be more pathogenic. It could be a real problem.
Yeah.
Maybe not.
Well,
But,
and
you
I was gonna say, Candida, isn't that the one where like you can basically starve from Candida, right? It totally decimates your digestive system, and you can't metabolize any food, and you end up in the hospital, and people fight for their lives on that. So this is something that could definitely be used today, but
Mhm.
could become way more helpful in the future too.
And how often? I mean, if you can treat it in hospital, yeah. But, like,
Sometimes people die.
Is it the kind of thing that's like prevalent up? Well, here's the
So
thing.
the issue is, Justin, is like it's not for the majority of people, and for the majority of people, bacteria, fungi, all sorts of things are not the big issue. But there is the percentage of the population that do have health issues that predispose them to having an immune system that can't fight off the fungus, that can't fight off the bacteria. And so these are the populations that the vaccine candidates are going to be most successful with and for.
Yes. And as long as the drug companies keep a narrow range of clientele as their goal and don't try to make it a widespread thing that everybody does because we are entering a back and forth battle between bacteria and fungi that have been going on for billions of years.
Yeah, I think that's Kitty's point, is we've been using antifungals for decades. So the vaccine is way more effective and way less likely to create more dangerous strains than antifungals are.
If antifungals are
waggy finger thing happening, as long as they test this very thoroughly before any widespread vaccination of anything takes place.
And we hope
I'm not sure we understand the fungal microbiome yet. And that's kind of my argument. Like, we're starting to get the bacterial interaction. We're starting to learn that in the last 5 to 10 years.
Agree with
We've
that.
been figuring that out. We don't really know the fungal economy as well.
We just know the bad ones, that when it's bad, it's bad.
Yes, we can start with the brain-eating funguses.
The fungi that we like to eat, or, I don't like brain-eating funguses. Nope. No, thank you. Okay. No brain-eating funguses today. This is This Week in Science. We are here with you to talk about science and to have a whole bunch of fun, and we hope that you enjoy the show. We hope that you enjoy it every single week, and if you would like to help support our show, that's how we support ourselves. Listener support is the way that we do it. You can head over to TWIS.org and click on our Patreon link, and when you get to Patreon, then you choose your level of support, $10 and more per month, and we will thank you by name at the end of the show. And I can't wait to read your name at the end of the show. Get your name on there for next month. But now we're going to come back. More This Week in Science. It is time for that wonderful part of the show that we love to call Blair's Bigfoot Corner. No,
With
Blair's
Blairfoot.
Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
What you know about the dodo?
It's dead.
It's dead. You're right.
Oh,
Yes.
we killed it.
In about
They were
1681
tasty.
is when the last dodo was killed. It was a combination of the fact that they were very tasty and too friendly, but also that pigs and other introduced animals brought by Portuguese sailors also thought dodo eggs were delicious. They were just too delicious for their own good. It's a big bummer. Well, Colossal's at it again. In March 2022, they sequenced the dodo genome, and now they have announced they will be de-extincting and rewilding the dodo.
Awesome.
Why?
So here's the deal.
Okay, thank you.
The asterisk to the asterisk to the asterisk on this, as it always is Also, this will help us further genetic resource tools focus on birds and avian conservation. So it's the big sexy fundraiser that got them $150 million that they're excited to bring back the dodo, but really we're going to use it to help endangered animals instead. So their intentions is to bring the dodo back, and this will be through genetic rescue techniques and the de-extinction toolkit. And I was like, that is vague. And so I did some more research, and I found out, first of all, that one of the researchers, a woman from Colossal, totally lost her name, but her last name is Shapiro. I know that. But she said, quote, "It would be disingenuous to say that we're recreating something that's 100% identical to something that existed."
Beth Shapiro.
Beth, thank you, Beth.
She's a lead paleogeneticist at Colossal.
Right. So yeah, that's the thing. What we're trying to do is create proxies for these species that are adapted to environments that are there today. So this was true with the mammoth too. They're going to be hybridizing it with current extant species. It won't be a mammoth. This will not be a dodo.
Well,
I hope they're not mixing the mammoth and the dodo.
No, they're not. They won't be doing that. Here's
the question, though.
Hold on, let me tell you how they're doing it. Okay. So they have many significant technological hurdles, ethical caveats, and unanswered questions. But this is their intention. I want you to hear how bonkers this is. Okay, so unlike with mammal projects, where they would genetically engineer sperm, egg, implant it in a kind of analogous mammal to mature, which, as we know, when mammals grow inside a mother, the DNA gets all mixed up and jumbled, and that also makes it less of a mammoth, right? In this case, since it's coming out of an egg, they're going to work with primordial germ cells. They are going to extract these germ cells from a bird egg, like a chicken egg. About a day after it's laid, they'll culture it in a dish. In the dish, they're going to make DNA edits to the germ cells to try to match the DNA sequence that they sequenced of the dodo until it becomes as dodo-like as they can. And then they will reimplant the germ cells back into the egg, and then they can incubate the egg.
Brilliant.
There's no way they're breaking anything doing that. Oh boy, that's a lot of edits. I'm sure.
Then the thing that hatches will be a chicken still. But its reproductive cells will contain some dodo-like cells.
Yes.
After fertilization, those cells might grow into dodos. So go ahead, Justin.
So, chicken and the egg, as we've discussed too many times already, egg comes first. You didn't have chickens before you had eggs that could make chickens. So, in the dodo, and actually even in the mammoth, your end result isn't that first hybrid.
Right.
Isn't that first elephant mammoth combo. What you're really starting with is the closest thing we could do by pure genetic alteration. And you're right, you're going to get a mix. Some of this, some of that, not the complete thing. But you still have more DNA that didn't take that you can add back in. You have, if there's a lot of elephant DNA in your new mammoth hybrid, next time around, you can do this again through your hybrid and get closer. So you can, you're not going to get it, that dodo, that very approximate dodo, the first run, but in subsequent generations, you can get much closer than you ever would in a first run, right? So this is, if your goal is dodo or if your goal is mammoth, starting with these iffy hybrids gives you a platform upon which to build to move that needle further in that direction, as evolution did in the first place when it moved things towards mammoth or moved things towards chicken or dodo.
And as they move things forward, they're going to be developing new technologies as investors are investing money in this biotechnology company. And what they're going to be doing is patenting a whole bunch of techniques and using that to be able to license technology out and make further money into the future, and possibly spin off other companies that will then hold the technology knowledge of how to de-extinct, how that is it is tagged to reproduction, that is tagged to this genetic health. And suddenly you have a group of companies or even a monopoly that are basically holding your genes and the future generations' genes hostage. Here we go.
Well, hostage. Look,
So-
we're in a situation where, what was it? 25% of the world's bird population is not threatened by global warming.
No. So
Yeah.
that
Yeah,
piece of that is really great.
We're at
because
a
you can
precipice
use
of
this- yes.
mass extinction, the 7th in the global history.
They
only the
are going to
7th.
make a killing off of this technology
Yes,
with all the things that are
absolutely.
going extinct. All the people who want to save this butterfly, save that bird, do
Yeah.
that.
Yeah.
How much money is going to be going into maintaining populations through
And
these
that
technologies?
is great.
Yeah.
But in terms of rewilding the dodo, I just
What
wanna
else are we
come back
spending
on
money
that
on, Kiki? Who is
and
wasting
say
it?
where. You know where dodos were found the last time we noticed them anywhere was off of a teeny tiny island off of the coast of Madagascar.
Mhm.
Do you know what it's like in Madagascar right now? Do you know what it's like in the
I haven't
islands
been
around
recently.
Madagascar right now? It's not good. The rainforests are disappearing.
Everything's
So,
disappearing.
Everything's going away. I don't know where these dodos are going. This really feels like they would be making these to put them in zoos. They would be
Yeah.
making them for the novelty, to be able to go to
Yeah.
come see the traveling
Yeah.
dodo exhibit that goes around. And
Yeah.
what is the point of that? It's
Well,
that's
they're
the part
certainly
that really
not-
bothers me.
Yeah, they're not going to be part of solving our egg crisis, our egg shortage.
No.
They were like one egg a year, like very slowly reproducing organisms.
But apparently delicious. I don't know.
Well, actually, the story might be the eggs were delicious. Maybe the actual dodos may not have actually tasted that good, but they were the explorers who first saw them. Had zero fear of humans.
Mhm.
Yeah.
So unlike other animals that would run away, they could
Easy to eat.
wrangle them all up and put them on the ship. And compared to basically the dry biscuit that sailors are forced to eat on a daily basis, anything was better than that.
There's a poem by Henry Carlyle called Dodo. Years they mistook me for you, chanting your name in the streets, pointing grubby fingers. Today in the Natural History Museum, I saw why, Dodo. You look the way I feel, with your sad, absent-minded eyes and your beak like a Stone Age axe, even your feathers, dingy and fuzzy. What woman would want them for a hat? With a name like Didus ineptus, where could you go? Wings too small to fly with and feet so large and slow. You were not very palatable. Men slaughtered you for sport. Hogs ate the one egg you laid each year. Sometimes I think I know how it feels to be scattered over the world, a foot in the British Museum, a head in Copenhagen, to be a lesson after the fact, an entity in name only, and that taken in vain.
Wow. So yeah, we got to bring him back. See now, Blair, are you with me? We got to bring the dodo back.
For what?
For all of us who feel like dodos in our daily life.
That's a bad reason.
That's a bad reason.
All
That's
right.
a
Do
good
you
reason
want to
that-
move on?
Let's
Yeah,
move
we can.
jet lag. We got some jet lag
Okay,
here. Yeah.
great. Because I'm tired of my jet lag. No. You know who are jet lag though? Female fruit flies.
What? Why?
Specifically after mating. This is a study from Cornell University. They found that seminal fluid protein transferred from male to female fruit flies during mating changes the expression of genes related to the fly's circadian clock, akin to jet lag. There have been many studies looking at sex peptide from fruit flies. I'm pretty sure I've reported on quite a few of them and how they impact fruit fly behavior, but there's a lot of question as to the exact mechanism. After mating, sex peptide has been shown to elicit increased egg laying, aggression, activity, and feeding. So let's say, okay, increased egg laying, great, good for reproduction. Aggression, great, keep the other boys away. Activity, yeah, you got to prep for the babies. Feeding, got to feed those babies. While reducing sleep and reducing interest in mating in previously unmated females. Once they've mated, they're not interested in any other men. So these very same behaviors, they were changed directly related to sex peptide, but basically they were also able to see those same behaviors linked to times of the day. And what they found was that one way that the sex peptide might be actually impacting all these different factors is by confusing the circadian rhythm of these female flies and shifting their internal clock. So they wanted to explore this, see the kind of mechanism going on. So they examined transcriptomes, which the RNA sequencing that reveals gene expression. So basically the mechanism by which genes are turned on or off at many different time points. And so they wanted to see the order in which things happen. In the first 4 hours after mating, they found the expression of genes involved in female fly metabolism and the circadian clock. They don't know what triggered these because it wasn't sex peptides, but they think it might have been pheromones or seminal fluid proteins. These were short-term effects. 4 hours after mating, the researchers discovered that sex peptides specifically caused changes to their circadian rhythm regulation and genes that regulate the circadian clock pathways. So the sex peptides specifically mess with the circadian clock 4 hours later. So basically, it was almost like a stopgap with some other mechanism. They think either pheromones or seminal fluid proteins, they're not sure. But then after that, the sex peptide kicked in, messed with their genes that impact their circadian clock. So in the future, they want to see how long the effect lasts and whether this happens in other organisms, since so many organisms have circadian clocks and the genes that control circadian clocks are highly conserved across the animal kingdom. So is this happening in other species? It's a neat idea.
I don't know, it's a really interesting question. And people talk about getting sleepy, want to take a nap after coitus. There's what other behavioral changes occur, and is it what about the short-term activation and then that long-term activation that's a little bit not permanent but a little bit more dramatic of an effect.
Yeah.
What does that mean though for the female fruit flies? So after it happens, that they're like, okay, now I need to go eat and go to bed. Yeah. And it keeps them away from other males, right? Yes. So they're going to be like, okay, I'm done. I don't need, I'm not active anymore.
That's the other question. Does this happen specifically in species with a lot of competition for females? Right. Yes. Does it happen all the time? Does it impact circadian rhythms in different ways depending on what things are like in that individual animal? Because flies, they're dealing with a very short lifespan, so every hour matters. And so you could see how it would be so urgent that they'd be like, no, this is your focus now. Leave all other men alone and take care of these eggs, and that's it.
Right. I mean, that's what about birds maybe who have a single partner? What about, say, cats who have multiple partners? What about, how do these different signaling molecules affect things differently? Or do they?
How about herds where you have one male and a bunch of females? It wouldn't be
Right.
very
A pride.
effective to
Yeah.
have them be aggressive around each other after mating, right? So, yeah, so that's the question. Is sex peptide impacting animals across the animal kingdom? Is it in similar ways? Is it in different ways?
I'm going to guess different,
yeah.
depending, because it's, you know, it's all going to be molecular pathways affecting neural pathways, and
Mhm.
you know, the timing is going to be important, and also what those specific pathways are.
And hey, if we can cure insomnia while we're at it, there we go.
There you go.
The best cure ever.
Yeah.
Justin, do you want to talk about seawater?
Yeah, so as we continue to wait for fusion energy that is 20 to 40 years away and always will be, has been for 70 years, one solution might be hydrogen. Maybe. Hydrogen economy for energy production, for all its promise of creating energy out of the most abundant element in the universe and leaving only water in its wake, is really not here, and is also in that category of 40 years away and always will be, it seems. So it's in the most basic form of this chemistry, hydrogen is heated or used electricity to create oxygen. Heated with oxygen. Sorry, hydrogen is heated with oxygen, forms a chemical reaction that forms water, and out of that chemical reaction, a large amount of energy is released. While there are hydrogen cars on the road today, it is by far the least practical application of this technology. There won't be hydrogen cars in the future. The volume of hydrogen requires even when compressed to 700 atmospheres, contains about 30% of the equivalent energy per volume of gasoline, and takes up only, it's a horrible. You'd have to have giant tanks of this that also need to be pressurized, so they're going to be very heavy, and they don't work in cold weather.
And then on top of it, the usual issue is that you need pure water. You need fresh water as opposed to anything that has anything else in it, because you've got these electrodes that can't take the crudding up that happens, and
So that's for the production of the hydrogen in the first place
itself. Yeah.
Right. So this is in the use of it as an energy. Just everybody who's thinking about hydrogen cars, forget it. We can use electric cars. That's going to be the future. And we can have hydrogen power plants that are supplying that electricity to the grid. So that's the first thing we need to focus on.
Yeah.
We need to change the scale of how we're thinking about hydrogen and how it's going to interact. So then there's the bigger problem that you're pointing out, which is how do you get hydrogen in the first place? Currently, nearly all, something like 95% of our hydrogen is produced using fossil fuels. Electricity,
yes.
From coal, natural gas, along with methane in the process, breaks apart the water to extract hydrogen in a process called electrolysis, which also relies on platinum and uridium as catalysts, which are themselves very expensive and rare, hard to get. So hydrogen isn't really so much a energy source at this point. Even when we are effectively using it, it's more of a transfer medium, like an expensively, awkwardly high pressure battery.
Right.
So and like you're saying, it typically requires pure water, which means deionized fresh water, which is also a finite and contested resource, or desalinated, heavily treated salt water that adds tremendous expense on top of everything else. So forget I mentioned it.
Because desalination, that's not easy yet.
Not easy, not cheap. So forget I mentioned any of this. hydrogen economy.
Okay,
is
you
never
didn't.
going to happen.
So we're done. The story's done,
Yeah.
right?
Oh, in a completely unrelated story, though, a team of researchers published a paper in the journal Nature Energy detailing a novel method for performing electrolysis on untreated seawater with a cheap catalyst. Basically, leapfrogging a bunch of giant hurdles for hydrogen. According to the researchers, it worked just as well as the existing high pollutant, expensive methods. They still want to do some more extensive testing to see how well their catalyst aspect holds up over time. One of the reasons for using all of this pure water and these really expensive catalysts is there's chemical reactions that take place, especially within just seawater, which current systems why current systems are using pure and desalinated water. But so far, they have not seen any of the normally precipitating formations on electrodes that are suffered by these other systems that then bog everything down and stop the process. If this technology bears out, it's a tremendous game changer.
Yeah.
If we could scale this, the hydrogen economy could actually, which has been rather dismissive of in the past, could actually become a thing. There's a few more advances in material science, problems with hydrogen under that much pressure. Maybe we could use less pressure, but hydrogen under pressure also tends to destroy the tanks that it's contained in at a very rapid rate. Those little hydrogen molecules, under pressure, get in everywhere and destroy things. So there's still some things material science needs to do to make this a replacement for fossil fuels, but it sounds like this research has done a tremendous amount of the heavy lifting required for finding a source of hydrogen that can be cheap and not reliant on fossil fuels to produce. And with that piece out of the way, the rest of the components in the line become much simpler. Granted, we need to get rid of the idea of hydrogen cars. We got to just, I know, I love the idea, but electric cars, hydrogen is just right now another storage method for fossil fuels. It's like the electric car that's charged up off of the coal-fired power plant. It's that same problem with, hey, our end result looks great as long as we only look at this piece. Right?
Yeah, we could rewrite that one old song. Hydrogen's just another word for using fossil fuels.
Yeah, very nice. Ooh, very well done. Very well done. Yeah. And my last story tonight.
What you got?
Oh, this is going to be a fun Neanderthal skull story. A cave in Spain has been found to contain a large amount of skulls gathered there by Neanderthals. So these aren't skulls of Neanderthals, but animal skulls gathered together in one place by Neanderthals.
Oh, that's where I left them.
Yeah. There's a interesting note is that there's a lack of teeth or jaw bones, and there's evidence of cut marks leading researchers to believe that the animals were initially rendered elsewhere, probably wherever they were killed, because these are big animals. And the skulls were brought inside with a secondary round of processing that then took place, possibly related to removing the brains and soft tissues in and around the skull. This behavior, they say, seems to have no subsistence-related purpose, but to be more symbolic in its intent. And whenever archaeologists say something has a symbolic intent, it's helpful to remember that that's just a way of saying they haven't really figured out why.
They were making a museum.
Yeah. Like I actually very much agree with you on this. So, Neanderthal sites are usually associated with very strict subsistence activities, hunting, processing for the consumption of animal resources, preparation of tools for the use of hunting or rendering animals, and use of fire for warmth or cooking. So there's not a whole lot of frivolous Neanderthal activity that we've noted. Occasionally, we've seen odd things collected, artifacts such as novel stones that aren't from an area. There's that one example, at least, of a seashell that came from quite a distance away that was found in another cave. This cave, discovered in 2009 during an archaeological survey of the area, which has some other Neanderthal and interesting old cave areas. It's a zigzag cave. It's 80 meters long, 2 to 4 meters wide, and has collapsed. It lost its roof through erosion some time ago. And in there are some Neanderthal remains. There's the remains of a young child. And there's evidence typical of Neanderthal flint knapping, tool making, and some pretty extensive fire use, including what seems to be a large fire sort of pit or fire area in the cave. The skulls that were collected were found adjacent to that fire area. And these are mostly ancient cows, extinct cows, bison and urochs that were European cattle
Oryx?
back in
Oryx.
Is it
Yeah.
Oryx? Oryx?
Oryx?
Aurochs. Urochs.
Or Oryx?
Oryx.
Yeah, it's Google how to pronounce it. Anyway, there were also deer skulls, but interestingly, all of the deer skulls still had antlers. So no female deers and no male deers that had lost their antlers previous to
Both
ending up
males
in the
and
cave.
females lose their antlers.
Is it? Well, lose their antlers. Okay, but these were all with the big male antlers and still attached, so they hadn't lost them.
Okay.
The largest skulls in the collection were from 2 rhinoceroses.
Those would be big.
Those would be big. And also, yeah, Spain had rhinoceroses 50, 60,000 years ago, which if you didn't know, now you do. So all of these skulls, what I found very interesting is they either had horns, antlers, or, of course, the rhinoceros with its nasal horn.
Single tusk. Yeah.
Mhm.
Well, not a tusk, a horn.
Yeah, tusk, yeah. But
It's a horn.
It's a horn. It is a horn.
It's
yeah.
a horn.
Very specific term.
The Danish word for rhinoceros is I think nasahorn. Nose, or they just call them nose horns.
The nose horn animals.
And this collection being approximate to the fire pit area of the cave would have given them a great ambiance in that flickering firelight. Researchers do some speculating whether these could be, I think their best theory is that these are trophies, maybe a hunting shrine, or may even have, they say, further symbolic, ritualistic, magically imbued meaning for the Neanderthals. I disagree. My best guess, keeping with the utilitarian Neanderthal behavior, is I think they might just be there for training purposes. This is the idea, the fact that they've only brought skulls that have defenses built into them. Neanderthals are thought to be close-up hunters, even though we have some examples of spears that could have been thrown. But they have to, by and large, get pretty darn close to their prey.
Right.
The fact that they're by the fire makes me think they did this practicing at night. Because if you're a Neanderthal, daylight's for hunting. Daylight is for hunting and gathering. Daylight means you got to go out there and start tracking animals and going out chasing or going on the chase. Go catch something. Got to eat. Got to catch something in the daylight. At night, you can't go out hunting. What are you going to do? Well, we got the fire to keep warm. Hey, it's also we can see. Hey, why don't we practice? Why don't we do a little bit of hunting practice? And you grab the antlers, and I'll make sure I can stay out of the way of them. And somebody else can charge with the big rhinoceros horn. We learn how to sidestep that or avoid that. I don't know. It seems like that is a much more realistic use for Neanderthals who, to their credit, maintained a technology of tool making over tens of thousands of years, hundreds of thousands of years, actually probably inherited it from Homo erectus for a technology of stone tools that's really millions of years old. Even the cave paintings that we've seen that have been attributed to Neanderthals look to me like a game plan for hunting. They look like if you were to strategize and be like, okay, we got one, you're going to wave the big stick and here's the animals running away from you. And so we're going to be over here hunting them. They all look like hunting strategy. So what better thing to do is to bring home some practice elements of your most dangerous prey.
That's a fair it's a fair hypothesis. And I think, you'd have to think about like theirs is the hypothesis that it could be this mysticism, spiritual purpose. There's could be more practical,
or hunting trophies.
It could be hunting trophies.
Look, we got this big horn thing.
Right. I mean, into the sense of it being more practical and less of a spiritual thing. I mean, what else are they finding? Are they finding other little trinkets? Or they find, it all depends on the context of what is being found together with all this. So maybe there'll be more of a story as they uncover more things, as they, take a look more into this cave and this area where they found the bones. But yeah, it's the big interesting aspect is that it's not a graveyard. It's something purposeful. We just don't know what. Yeah.
It's too bad there's not cave paintings next to all of them identifying what they are and why they're there.
Yeah, we're like in a little box down and to the right of each one.
Yeah.
But yeah, I mean, in that sense, though, it would be like a museum collection as well. And again, keeping it, the fact that it's by the only light source in the cave makes me think that they spent some time appreciating them aside from just a collection of trophies. I think they played with them.
Right.
They might have been toys for the kids.
Could have been. You don't know. But as Harjar in the YouTube chat room is saying, you clean them by the fire and toss the bones. But if there were more bones found, then it would be more of a garbage pile.
Right. And so that's the thing that's also the thing they're pointing out is that there is not evidence of the rest of these large animals being rendered there.
Skulls.
And the fact that the skulls are typically left behind when you've rendered an animal and taken the meat.
And these
it's
are the big
heavy.
animals.
It's a heavy thing to lug around a rhinoceros head. It's
They're
not
a pain to clean also.
They didn't save the squirrels and the bunnies.
Yeah, and the fire may have helped do some of that cleaning too. You cook a skull a little bit to get off all the little.
Yeah, but the brains are so hard to get out without a Dremel.
Yeah, and there is some evidence of some puncture wounds that they think that they used to remove the brain matter.
Yeah.
So
because that's going to be very nutritious.
And also stinky if you don't get it out.
Get it out.
But I think in the bottom line here is that Neanderthals had to be good communicators and teachers to maintain the technologies that they used we don't have a language on this planet right now today that's 20,000 years old. 30,000, 40,000, 100,000 years old. We don't have anything like this. You needed a way to communicate and teach, probably verbally, but also with anything lying around. If you were
sticks and skulls and bones,
especially if you imagine a limited language and having to see somebody make a stone tool and observe it in order to know how to do it yourself, then absolutely these skulls with antlers that you're waving around is how you would have to teach the next generation with visual aids. You couldn't just explain it in enough detail using language if the language is primitive, which it may not have been. It may have been very sophisticated.
They may have had a very nice Shakespearean accent, too.
They may have.
Yeah. Well, talking about brains, I have a brainy story related to, not how we teach the brains, but brain development researchers just published in Science magazine this last week their work on the mitochondria in neurons.
You talking about the powerhouse of the cell?
The powerhouse of the cell? Yes, I am. I'm telling you about the powerhouse. This powerhouse is very important because it supplies the energy for the metabolism for the cell, and so it allows the growth of the cell to take place at a certain rate. So these researchers who have published this story in Science, they had been working on an idea of neuronal growth based on just some intrinsic property of the species it came from and the neuron itself. And so mouse neurons, they grow a lot faster than human neurons. And we've had this whole conversation about how human brain development, it takes a lot longer. And so that's part of how, we have the cognitive processes that we have and why, we have the intelligence that we have that, we don't necessarily see in other species because we're not looking carefully enough. But the researchers had looked at an idea that maybe the mitochondria are involved in the development somehow. And so these researchers had to figure out how first to basically mark a neuron when it was born so that they could keep track of it and be like, okay, you were born and now you're this old. And so basically you have a clock to be able to figure out how old a cell is in the brain. And so they incorporated a promoter region, a genetic element called NeuroD1 into some different cells. And they looked at respiration rate of the cells and the oxygen intake of mouse neurons and human neurons. And they found that when they changed the oxygenation rate of the cell, that they were able to promote the metabolism of the mouse neurons so that the oxygen consumption rate was like 10 times that of the human neurons. So they determined that the mouse neurons are using more oxygen and they're respiring more quickly than human neurons. And so then they were like, aha, let's look at the mitochondria. And so then they slowed down the mouse mitochondria. They sped up human mitochondria, and they were able to get neurons that developed at the exact same rate. They were able to implant mouse neurons with human cells and get the mouse neurons to grow at the same rate as the human cells. And so this could potentially be one of those sticking points about, if you stuck a mouse neuron into a human brain, or if you stuck human cells into a mouse brain, why don't they match up entirely perfectly? Well, this could be one of the things that allows it to happen. So the mitochondria comes down to the mitochondria. Now, this doesn't necessarily, give us the underpinning of our intelligence, our cognitive abilities, all that kind of stuff. But understanding how the mitochondria are involved in the development of neurons could also help us understand neurodegenerative disorders. It could help us understand some developmental disorders where neurons don't grow at the appropriate rate, and we haven't been able to really figure out why. And so the researchers are just focused now on looking more specifically at these neuronal mitochondria and figuring out how they're involved in the health and development of the neurons and the brain.
Yeah, because if we can find, if we end up finding that there's a problem with mitochondria and neurodegenerative diseases, then we have finally a target.
Yes, and there have been other stories that have suggested that mitochondria malfunction are a part of aging disorders of the brain, and that there are other specific, like mitochondria. There have been a lot of studies over the last few years that have really started to pinpoint mitochondria as a very important aspect of the neurons, which are the subunits of the brain. Yeah. Oh, and for anybody out there who is interested in vitro fertilization, a study came out this last week in the journal Molecular Human Production about the dangers of alcohol use.
Wait, what? There's dangers of alcohol use? What are you talking about?
So this is in the way that we talk about pregnancy and conception, it's all on the woman.
Yes.
Ladies, do not drink. You must change your diet. You must be
No hot tubs.
No hot tubs. You must be the pinnacle of health. And you be perfect. And then you will be able to conceive. And if you
exercise, but not too much.
But not, you don't want to bounce up and down too much. But anyway, the problem is that we've been focusing on the female side of the IVF question for a very long time, and
I am shocked.
Yes. Yeah, I thought you would respond that way, Blair. This is why I was like, I got to bring this story. So researchers have recently started looking into, okay, what if we started looking at the male side of the equation? So we say, hey, ladies, eat well and don't drink. But what happens to the chances of successful IVF if the man is drinking? So they did a study.
I don't like where this is going.
They did a study not in humans, in mice. So it's a mouse model, not a human model. So that's a big caveat here. But they pretty much had the mice in an abstinence from drinking group, a group that drank the legal limit of alcohol. So they were drinking like a 6% body weight or whatever alcohol. I don't know. And then they had a group that was over drinking and were getting drunk, like 10% alcohol blood levels. And they let the mice drink for like 6 weeks. So there's a part of this that I'm like, you just let them drink? For 6 weeks, like how were you controlling when they had the alcohol or not had the water and had water and anyway.
There's so many variables there. That's crazy.
Well,
not to mention how long
here.
6 weeks is for a lab mouse.
Yeah, but
6 weeks for a lab mouse. But so it was chronic. It was a chronic voluntary. They volunteered to. It said it in the voluntary like over drinking,
right.
binge
so
drinking.
What they did is they gave the mice
drinking.
they gave these mice dead end jobs,
right?
With and had them living paycheck to paycheck, and the rest took care of itself.
Yes, exactly. And these mice who drank any amount over the 6 weeks were less likely to have success in the in vitro fertilization. So they took the sperm from the mice, they stored them, they transferred them to embryos, and then looked to see what the rate of conception actually ended up being once female mice were implanted. They found that preconception paternal alcohol exposure disrupted embryonic gene expression and 2 critical regulators of the trophectoderm stem cell growth and placental patterning. This is FGF4 and EGFR. And there was lasting impact on the histological organization of late-term placenta. So there's placental histoarchitecture that was affected. Also, altered regulation of pathways that controlled mitochondrial function, oxidative phosphorylation, and some other imprinted genes. So which genes are getting expressed and how. So there's a take-home message here that perhaps all the this is mice, like all these caveats still stand. This is a lot of alcohol for the mice. This is mice. This is not people. But if you are in a situation where you are, you're spending money on attempting to conceive, and you want to give yourself the best opportunity as possible. So perhaps there should be messaging for the men to live that wonderful lifestyle similarly to the women in those relationships
For 6 weeks.
For 6 weeks,
right? Well, but for humans, that's like months. But anyway,
Yeah.
I think the thing that's really interesting here is when you talk about fertility tests in humans, in men, it is always sperm count related.
Yeah.
Does whatever
And
the man is doing impact sperm count? There's a little bit of conversation about sperm quality, but that's all this other stuff you're talking about. The idea
That's quality.
that's impacted.
That's quality, right? That's going to be, that's not necessarily count. So if all these mechanistic factors of gene and like placental production are being affected, that is quality.
Right. And
Not
I feel like
necessarily
a lot of
quantity.
the sperm quality conversations in human fertility is like motility, shape, like all these other things that are not directly related to what could happen late term. Like the fact you're talking about late term placenta stuff, like that's so far down the line past just like, did the sperm make it to the egg okay? Which is really what it's always been about before.
Did it make it?
Did the package make it to the destination? After that, it's up to mom, right? But it's not. This is proof that it's not. Which it's 50% of the DNA.
It is 50% of the DNA, and there's a lot of not just the DNA, but also RNA, very small molecule RNA that is also packaged with the sperm that has impact on a lot of genetic factors and expression. And so, and those are the small molecule RNA are going to be more likely to be epigenetic and based on recent environmental exposures. So stress or poisons.
Well, now you say poisons, but doesn't the dawn of humanity, when all of our great achievements started taking place, didn't that sort of correlate with our alcohol use?
Yeah, we just need to chill out.
Likely.
It's
everything's about
everything in moderation.
Might have actually been an accelerant.
The other thing I'm thinking about now is
chemical induced.
If you loop it back to my fruit fly story, you have the genetic package of what is in the sperm and the quality of the sperm, but we also do not yet know about the impact of seminal fluid quality, or what would it be like potential problems with seminal fluid as a result of behavior, right? So it's the more we learn about kind of the holistic mechanism of the part that the male has in that, I'm very curious to see if in the future, if we're going to find out that like the hormone releases directly after fertilization due to the proteins in seminal fluid can have an impact on fetal it's possible, right?
It's possible. All this stuff. Yeah.
Yeah.
I mean, there's no reason.
So, but we tell men, go nuts, do whatever you want.
Do whatever you
just
want.
The woman has to worry about
take care of
so
it.
That's is that true though? Because I feel like we've at some point covered or talked about this, that there's a 6 months. I was joking at 6 weeks before, but 6 months before conceiving, if you're trying, that the men should also be taking their vitamins, avoiding devices. Like I've always heard that.
So it depends. It depends who your doctor is.
Yeah,
it depends who your doctor is, because there's not, the jury's out. There's still research coming in. What I hear most from all of my friends of childbearing age that are going through this is that the man might be told also not to go in hot tubs for like maybe a week before you try to conceive. But sperm is created so often that like a lot of it is understood that the long-term stuff that something you did last week, that's not going to impact your sperm. You have new sperm now. But it's that can't be true. Your body is a system, right? And other than that, like until you have trouble conceiving, most of the time men are told, do whatever you want. You're not even thinking about it. Yeah. Do what you want.
I have 4 kids and they all came from just hugging somebody for too long.
Okay.
Like all of my kids were
I'm learning something interesting about Justin.
Yeah, not accidents. Okay. Because if you figure out how babies are born, you figure out like, oh, okay, well now that's how that happened. But there wasn't a whole lot. I'll just say there wasn't a whole lot of pre-planning involved in any of my wonderful,
Right.
beautiful humans.
Right. And so, and it all depends, but this specifically was looking at the situation of in vitro fertilization. And if if we're going to be talking about, all of the things that a woman has to go through to make that possible, there are also conversations that should probably be had that relate to, like Blair said, the quality of the sperm, not just the quantity, and different things that men can do as well, that partners can do in conjunction.
And it is
All
interesting
right.
because it also, just the last note is, when you have a pregnant spouse as a man, you often become somewhat of a support animal in the whole process. and so if you're really planning to get pregnant, and this is a thing that you're having to take the extra steps to try to achieve, you've already walked into the territory of support animal. So even if in the back of your mind, this doesn't make a difference or this doesn't matter, that doesn't matter anymore, because you're now a support animal and just do everything that you're being asked. That's how that works.
Be a partner. Be supportive. Yeah. It's a big step. Like it is ending this show. Sometimes it's a very big step from one to the next. We made it. We did it, right? All the stories.
So.
We got
That's
through it. Thank you, everyone, for listening. I hope that you all enjoyed the show. I would love to give some shout-outs to people who help with the show and make it possible. Fada, thank you so much for your assistance in show notes and show descriptions and social media. It's so great to have you working on that. Gord and Arnlore, others, thank you for keeping our chat rooms nice places to be. Identity 4, thank you for recording the show. And Rachel, thank you for your editing assistance. And of course, I would love to say thank you to our Patreon sponsors. All of you on Patreon, you're amazing. Teresa Smith, James Schaefer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Derek Schmidt, Don Mundis, Steele, Vin Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Aronovich, Kevin Reardon, Noodles Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leastman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Chemi Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Aaron, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if you would love to support us, head over to TWIS.org, click on our Patreon link, and you can choose your level of support. Woof, on next week's show.
We will be back Wednesday, 8 p.m. Pacific time, and again 5 a.m. Thursday, Central European time, broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Want to listen to us as a podcast? Perhaps while you sit in the hot tub, if you're allowed. Just search for This Week in Science where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to stories will be available on our website, www.twis.org.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into a syringe that gets injected into a chicken egg. They'll mess with the DNA a bunch, and then nothing else will happen. It'll just sit there, and we won't read it.
It'll be broken.
And if you want us to read anything you send us on Twitter, hurry up. Currently, we are at TWIS Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. But we're Also on the, do we have ats or hashtags for the Mastodoniverse
TWIS
yet? We gotta get
Science.
those added in. At TWIS
Yes.
Science on the Mastodoniverse server for the Mastodon, I think is how it's going to go. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.